Okay, s- so let's get started. I'm just, yeah, okay. So let's get started. So today, it's mostly about it's about the course recap. So I'll recap from the beginning, from graph theory, what we started, and till the end of modeling, what we covered everything. I'll just give a brief overview of the whole course recap. And before that, we'll discuss a little bit about robustness and evolvability, and how you can how these two things can coexist and in biological systems. So, okay, let's get started. So first comes the concept of neutral networks. So what are neutral networks? These are basically, first you consider a space of discrete macromolecular sequences, which are basically genotypes, uh, which can be RNA or proteins. And in this space, two sequences are immediate neighbors if they can be transformed into one another by a single mutation. So, for example, it's one sequence is ATGC and another sequence is ATGG. So, changing the last nucleotide using a point mutation, you can go from one sequence to another. So, these kind of sequences will be neighbors in neutral networks. So, these are immediate neighbors, and if you can go from one sequence to another by a single point mutation. And a genotype network, which is the neutral network, which is called, it's a connected set of these genotypes which set a single phenotype, which can give you a single phenotype. So you have a set of genotypes which are connected to each other due to the fact that they are close neighbors to each other. They can be RNA sequences, they can be DNA sequences, they can be protein sequences. Whatever phenotype you are trying to match, those particular genotypes, so those sequences which give the same phenotype, those are mapped together. So that mapping of various different genotypes, various different variations of genotypes giving the same phenotype is called a neutral network or a genotype network. And within the genotype network, genotypes are directly connected to each other if they differ by this small mutational change. In maximum cases, this is a point mutation, but you can also consider uh, bigger mutations. I'll not go to the level of structural variations. Maybe you cannot consider those, but even like 5 or 7 base pair differences, you can consider. So you need to set up your network in that fashion. And genotype networks are thought to be common organizational property of genotype spaces of biological systems at different levels. And ample evidence for their existence supports this notion for RNAs, proteins, and binding sites of regulatory proteins. So your genotypes are your sequences, uh, basically, and if you can map these onto RNAs, proteins, or binding sites of regulatory proteins, and you can use this as a map, genotype phenotype map. We'll talk about what these are and how these are important. But essentially, you have a set of genotypes and you have a set of phenotypes. You want to map them. That is your ultimate objective. And the exploration of such networks became only possible with the ability to characterize molecular phenotypes for many different genotypes, either computationally or experimentally. So if you want to construct this kind of network, the information that you most require is basically which genotypes, which variations give a same phenotype. So that information that needs to be either experimentally obtained, you can do basically molecular biology, you can use molecular biology techniques to find these variations which are giving the same phenotype, meaning you will mutate and you'll see no change in phenotype. So uh, these uh, these type of genotypes which uh, differ by small amounts and give the same phenotype. These are your these are the basically the which form the genotype network, and the phenotype which may be protein folding, which may be a certain phenotype that you are seeing outside of, because directly from genotype uh, to the outer characteristics, it is the mapping is uh, very difficult. So for example, if I want to map DNA variations with, say, for example, hair color, so that mapping is very difficult because you're going from one end of the spectrum to the other end. So genes and DNA sequences to become the finished phenotype product. But this mapping is much more difficult than if you can consider the genotype network of your genetic variances, and you consider the ranges of proteins that are formed as your phenotype networks. So the mapping between this genotype network of your genetic variances and your phenotype network of your basically say a certain protein expression or formation of certain protein. So this is much easier than mapping genetic differences or genetic variations to like phenotypes that are not directly dependent on the gene, but also other regulatory mechanisms come into play in those cases as well. So you need to keep those in mind. The concept was actually originated by this a pair of uh, scientists known as Maynard and Smith, and they were developed later by some other collaborators. And they showed that neural networks associated with many secondary RNA secondary structures and are vast and span the whole sequence space. What they are trying to say is basically for seems RNA secondary structures, there are a lot, there are various genotypes that give the same secondary RNA secondary structures, and uh, these are associated. The association is very uncanny. So it, this is a very good phenotype genotype map that you can explore. Your phenotypes would be RNA secondary structures, and your genotypes would be your genetic variances. The concept was later applied to proteins and to higher order molecular systems such as regulatory network. So you see, the systems they are applying to are much more like in hierarchy, much more lower in nature. So they are not trying to map from a genotype to a say a eye color or a hair color or a skin color, something like that. So the mapping, if you want to have a good mapping, or your genotype and phenotype must be there should not be many layers between them. So then the mapping is good. So this is an example of neutral network. So here, each of these rectangles here, these here and here, each of these rectangle represents basically these six rectangles basically represents a part of space of genotypes, and these gray circles here, these corresponding to individual genotypes on a neutral network. So the idea is these are all so the individual nodes are sequences or genotypes that are connected to each other. If two nodes are connected to each other, they are very similar in nature. So maybe a point mutation or two or three base pairs difference. And all of these are connected, which means that these can give a same phenotype. Basically a straight line linking these two genotypes, and if they a straight line links the two genotypes if they can be interconverted to a single point mutation. So as I was saying, and these top three panels show how neutral evolution explores genotype space for a phenotype with low robustness. So in this case, the robustness of the phenotypes is very low. I'll talk about robustness a little bit later and how you can get robustness from these kind of networks and these kind of GP maps or genotype phenotype maps. But yeah, the this is a phenotype, the small neutral network in which individual genotypes on an average have a few neutral neighbors. So if a particular genotype has less number of neighbors, then it means it is not very easy to go from that genotype to the its neighboring genotype. Say for example, you want to go from this genotype to this genotype. Sorry, so these two genotypes you see, you want to go from this genotype to this genotype. Now these are not connected. If they are not connected, you have to go through all through here, from here, then here. You come about in a round, and then you come to that particular genotype. So although these look closer to each other, but they are not connected. So individualistically they are very different as genotypes, and if you want to go from that particular genotype to the other genotype, it's a longer process than being directly connected. And although these are uh, what to say these are separated up by a little bit. If you want to move to one genotype to another, you have to travel a little bit, but all these genotypes are part of the same neutral network because they give the same phenotype. That is an important thing that to keep in mind. And these bottom three panels show the same evolutionary process, but for a robust phenotype. See the difference between the first three panels and the next three panels are the number of connections between each node. So if you want to go from one node to another, it is much easier in these panels than these panels. So it is because the number of connections is much lower. So higher the connectivity of your neutral network, higher is your robustness, because it is much more easier to move from one point to another without affecting the phenotype. So, which is basically the definition of robustness. So, in robustness, if you want to test your system for robustness, your idea should be, if you perturb it a little, does it change its phenotype? Does it change its state, or does it remain the same? In the second case, since these are very well connected, so if you, even if a small change will not bring about any difference, because ultimately it is giving the same phenotype, because these are neutral networks. But in this case, if you moving from this point to this point is much difficult than in the lower panel, because they are more connected. So if your system has higher robustness, if a particular biological system has higher robustness, it is very much plausible that the neutral network of the genotype phenotype maps will be much more connected. So here the blue circles here corresponding to individual members of population, and the red stubs here, these indicate basically hypothetical deleterious mutations that cause a mutation to genotypes lying outside the neutral network. So basically, these red things are showing, what they're showing is basically these are deleterious mutations, which is the cause of which is due to these genotypes. You get it? So these genotypes are causing a deleterious mutation in another genotype, but that is not part of the same neutral network. That is lying in another neutral network. So that particular genotype phenotype map is not getting affected, but another neutral network is getting affected in this case. And differences number of red stubs between the top and the bottom panel illustrates that robust phenotypes with large neutral networks are subject to fewer deleterious mutations. And the two leftmost panels show that genetically identical populations with moderate genotypic diversity, in which several populations have the same genotype. So these basically the bigger your neutral network is, or what to say, yeah, the more connected, or the bigger span of your neutral network, if it is bigger, then what it means is basically, it's intuitively what it means is these are if you compare these two, these are like genetically identical populations with moderate diversity, because you know if you uh, if you want to map from your genotype to phenotype, uh, the diversity point comes in, if you're moving from one neutral network to another, your phenotype would change. So the bigger is one of the bigger your particular neutral network is, the lesser chance of you is to move from one neutral network to another, because just by probability it is more likely that you'll stay in that network and not affect other networks, as in this case the red stubs that you see here. So that is because it is less connected and it's moving to another neutral network, which is much lower if you can span your genotype network in a bigger fashion, and if it is more connected. Okay, so next comes the concept of this neutralism and selectionism. So in evolution there's a concept of selection uh, where nature selects for the most fittest individual. Sorry, huh. But uh, due to yeah, nature selects the most fittest individual. But how do you how does the individual get fit? It has to undergo certain adaptation, it has to undergo certain evolution, so that it can stand out from the rest of the group, and those particular traits which can help it live more, or help it live better, or reproduce better, uh, those traits will be selected, and the traits which are not as helpful as these traits, those will be eliminated. But in order to select for these traits, you need to evolve. You need to change your phenotype. You need to change your genotype. You need to basically you need to move from one state to another in terms of your genotype or your phenotype, whatever be the case. But here comes the uh, issue. When you're trying to define robustness, you're saying if your biological system is more robust, the better it is. If you perturb it a little, it doesn't move from its state. That is a better condition than if you perturb it and it moves from its state. But in case of evolution, you want that transition. In case of selection, you want the transition. And the importance of this robustness and this neutral change, why robustness and neutral change, robustness and selection can coexist and help each other, because these are like kind of antagonistic ideas. So for evolution would prefer changes, robustness would not prefer changes, but these two coexist. And uh, how they coexist is a major question. The importance of robustness and neutral change is highlighted by the studies of molecular phenotypes. Now let's consider a question. How do you reconcile these observations in the form of a conceptual model centered around the neutral network metaphor? So you have the selection, you have your robustness. Now you need to build actually a model to show how these both of these co-work and how like different variations are brought about. Say for example, for simplicity, you take a single genotype that undergoes random mutational change, which is basically a random walk of the neutral network. So, and you take any of the genotypes and you randomly walk depending on how it is connected. Assume the phenotype is suboptimal and the target of the evolutionary search is a better and optimal phenotype. So, the current neutral network you are in and the phenotype that it is spanning, it is kind of assume a suboptimal phenotype, and you want to move to a better phenotype, a more optimal phenotype. The sequence might first take several neutral mutational steps on the neutral network. Just focus on the neutral change, although deleterious changes are more likely in like real biological systems. It is more likely that you'll have a deleterious change, which will result in a loss of function or something like that, than a single point mutation every time. So but let's assume that this only happens that the sequence takes a single point mutation across the genotype map, and the changes are neutral in nature. And after some steps, what will happen is a phenotype changing beneficial mutation might produce a newer phenotype which is closer to the target phenotype. So what will happen is, just by doing this random walk, it will go to one point where it will basically move from one genotype network to another genotype network, because a phenotype and different phenotype will be formed. So this is what they're calling it, a phenotype changing beneficial mutation. And what will happen is this new phenotype that is formed will be selected because this is a better phenotype than the suboptimal older phenotypes that uh, we were seeing. And the random walker will thus hop from the first to a second neutral network. So as I said, what you are doing is you are in that particular neutral network, and suddenly you are doing random walk, and while doing that, at one step you moved from one genotype network to another genotype network. And so uh, this cycle goes on in evolution. This cycle goes on. So a number of neutral mutation and exploration of current neutral network would be followed by a mutation that discovers a newer phenotype closer to the target. I'll just explain it with a figure here. So for example, this is uh, um, um, genotype phenotype network, and this these gray circles are basically individual genotypes, and if they are connected, they are basically uh, one mutation away from each other. The genotype evolves towards a hypothetical adaptive phenotype. So basically, if you are moving here, 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 so these all movements are neutral. But the moment you move from this node to this node, this is a different phenotype altogether. This is a beneficial mutation, but the phenotype is different. But you have reached here through random walk only. It's not that you have followed a certain path to reach here and then you have moved to a different phenotype, a more a better phenotype. This is the concept is you are moving randomly. First, you are experiencing neutral mutations, and then at one point, suddenly, at the end of that network, you'll see that you're moving from one genotype network to another. Uh, the newer genotype network will be associated with the newer phenotype, and in order to arrive at this particular phenotype, in this case it traverses four different neutral networks. Is colored like these, and uh, these four different neutral networks, and uh, within each neutral network, evolution is neutral, but at the transition between neutral networks, positive selection occurs. So this is a network, and this is another network. So when you're moving from this network to this network, this is a transition. Now you're moving from this network to this network, this is also a transition. The rest of them are neutral mutations, doesn't make a change in the phenotype. So how does this explain evolution? So the evolutionary concept is the selection will be of the best traits. Now, how do you get that best traits? The model states, okay, you have a genotype map, you move from one genotype to another, you make neutral changes. But at certain point, what you'll do is you'll move from one genotype network to the other. If the second genotype network is an optimal genotype, then maybe you'll stay in that. If the second genotype network is corresponding to optimal phenotype, then maybe you'll stay in that genotype network. If not, if if evolution does not select that trait, then maybe you'll do the random walk again and find a new network which is beneficial, which will have a more beneficial phenotype than the older ones. Okay, so yeah, so this is what I've summarized here. So these are what genotype phenotype maps are. So this lower plane is your genotype. That can like in, meaning in the genotype map there can be like spaces for different populations, and this is basically your y-axis is showing the phenotype, and this plane that is getting formed is basically your genotype phenotype map. So which genotype will corresponding to which phenotype. So here three maps are shown. Basically these are three-dimensional maps. So so the entire surface represents a hypothetical global genotype phenotype map, relating all possible genotypes to a phenotype. So this is if you collect all in an organism or in a cell, take all the genotypes and you try to map it to a phenotype. So what you'll get is your x-axis or your basically xz plane will be your sorry this is y, this is x. So the z-axis should be in this side. So picture is a little bit misleading, but anyway. So uh, your one plane will be your genotype plane, another plane will be your phenotype plane, and the projection of these two on another one will be your genotype phenotype map. And the genotype phenotype map concept can also be extended to the description of physiological development and functional mechanisms by which genotypes detect the epigenesis of phenotypes. So basically, newer phenotypes getting formed, uh, evolution occurring, those things. And populations evolve by moving in the genotype space, which may change the phenotype and the structure of the local GP map, as we discussed earlier. In this figure, the global map shows a discontinuity in phenotype space, uh, such that um, in this figure, it shows a discontinuity in phenotype space as the genotypes on either side of the boundary labeled structural changes can show a discrete difference in phenotype. So like these genotypes, if there's a discontinuity in your genotype phenotype map, what it actually means is those particular genotypes do not correspond to a particular phenotype. So those mutations or those particular variants are maybe loss of function variants, or maybe a mutation which is not producing any kind of phenotype or not mapping to a particular phenotype. So yeah, these are genotype phenotype maps, and this is an example of a genotype phenotype network or a genotype phenotype map in case of sulfur metabolism. So uh, this the this is basically in this case the phenotype that they have considered is the flux balance analysis of a metabolic network. So it's the flux values basically. Your phenotypes are the flux values. Your genotypes are your basically your genotypes only, and what trying to map the particular genotypes to the particular fluxes. So yeah, each genotype on the genotype network has around 1221 neighbors, and they differ by one single mutation. And these neighbors are connected by edges, as we all know, of the genotype network. If you have a single mutation, you connect by edge. And two hypothetical phenotypes are described here, this here, as binary vectors listing the environment's genotype is viable in D. Excuse me. Random evolutionary walks can be seen as paths on a genotype network that stay on genotypes with the same phenotype, and these mutations correspond to additions or deletions of individual reactions from the metabolic network. In this case, it is the deletion of a metabol deletion of individual reactions, not a particular single sequence, but yeah, but the individual reactions from a metabolic network. It will be sequences if it were like kind of a gene network. If it is a regulatory network, it would have been transcription factors and gene. But yeah, so this is how the genotype phenotype maps looks like. So here is your list of genotypes. Genotypes is your list of pathways here, and you set them as vectors, do flux balance analysis, and this is your phenotype. This you're trying to map this phenotype to a particular genotype. So your ideal genotype phenotype map would show the, say for example, you are trying to maximize the production of this sulfate here. So what your phenotype map will show you is which genotype is corresponding to the maximum production of sulfur, because in this case you are doing FPA and you are considering flux, so you're trying to maximize the growth or maximize the production of a particular compound, and what you're trying to do is map that to a particular genotype. Okay, so these are different, um, just to give an overview kind of a thing, these are different kind of elements or factors that affect robustness. We have discussed some of these before, but just I'll go through it again. So modularity, which is the degree of interconnectedness in a system, is important because it mediates between this evolvability and robustness. So modularity is something that mediates between these two things, and this evolvability is facilitated by genetic networks of hub and spoke type. Basically, uh, you have you have different hubs, and the spokes represent the edges connecting those hubs. And, and this is based on feedback regulatory systems, which comes from robustness. Now, these two are inversely related, uh, but for for the mediation of modularity, these are something that go hand in hand. Say, for example, now natural selection, if you consider natural selection, at least indirectly affects evolvability. It certainly does. Now, natural selection indirectly affects robustness as well, and natural selection can also shape modularity. Now, there are a lot of these moving parts which can affect each other. I just wanted to show you just that genotype phenotype maps look cool, but the causal reason, the causal variant or the causal reason for a particular phenotype to show, it is very hard to find because of these various moving parts. Because like, see, natural selection affecting modularity. Now, modularity affects evolvability and robustness. Robustness is also affected by natural selection. Evolvability is also affected by natural selection. So there are different moving parts which regulate each other. So robustness as a concept is is very nice. It's very useful to create networks or create models where you want a particular outcome no matter what the perturbation is. But there are a lot of moving parts to be considered, especially for biological systems. So that is the reason I keep this plot, just to give an idea. Okay, it's not only about a genetic genotype connecting a phenotype. There are other parts of the system as well which are affecting each other. Okay, now this is all that we have for this week. Next, I'll go into uh, this recap. So I'll start from the graph theory concepts. till the modeling stuff that we have done. Let's get started. Okay, so everyone remembers this. These are like different types of graphs. So this is an undirected graph. Both are undirected graphs. So if you see D, it starts from D. Okay, this is an undirected graph. This is a directed graph because edges are showing directions. Uh, F is what is known as a semantic graph. So your edges will have some values and which are connecting one node to another, and these basically so and the relationship is basically more of a more of an intuitive relationship than a mathematical relationship. Uh, this is a weighted graph since the edges are showing weights. So basically, this is 3 and 4 is a stronger connection than 3 and 2 maybe. It's 0.1 and 1. And this is a bipartite graph. So you have two clusters of nodes. This 1, 2, 3 is a cluster. 4, 5, 6, 7 is another cluster. Now, cluster 1 connects to clusters 2, but individually they are not connected. So 1 does not connect to 2, 3, or 4 does not connect to 5, 6, 7 or something. These are the clusters are connected, but individually they are not connected. This is what is known as a bipartite graph. Uh, this is basically a mixed graph because it has edge weights, it has directions, depending on it. It is a mixed graph. J is a multi-edge graph. So what does a multi-edge graph mean? Meaning between two nodes there can be multiple edges connecting, and those can mean different things. So this is a multi-edge graph, and this is a hypergraph. So the edges that originate, they do not originate directly from the node; they originate from other edges. So this is kind of a reaction, metabolic reaction graph that we see. Okay, and this is basically a tree graph which is showing the hierarchy in case of maybe in gene expression. Okay, so connectivity, I hope all of you remember. So connectivity is basically how many edges are going in and coming out of your particular node. So in undirected network, your connectivity or your average degree, what no, the degree is defined as the number of links to other nodes, and the average degree is defined as is given by 2L by n, and in directed networks it is given L by n, because there are two components, k in and k out. Your in degree and out degree, there are two degrees in case of directed networks. Uh, in Yeah, so next are the paths and distances in network. So one is the characteristic path length, which is basically the number of edges in the shortest path length between two vertices averaged over all the vertices. It measures the typical separation between two vertices in a network. So this is your this is basically a path, but this is not the characteristic path length. Uh, if you Yeah, it can be a characteristic path length depending upon your approach. So this is what I mean to say is this is not the shortest path, but this can be the characteristic path length, and which can measure basically uh, all the network's overall navigability. And it is defined as the shortest path between two vertices averaged over all the vertices. And the network diameter is it's a similar concept. So you move from one end of the network to the other end, and what is the distance on how many edges did you have to travel. So it gives you about the span of the network, information about the span of the network, how big it is, or how spread out it is, basically how different is the extreme node at one end to the extreme node at another end, or how far away it is. This is what it tells you. Next is the concept of clustering coefficient. So this is basically the number of total number of connected subgraphs inside a network. So with number of triangles here denoting the smallest subgraph is basically smallest subgraph is basically a three node graph which are all connected to each other. And the clustering coefficient is given by the number of triangles connected to node I divided by the number of triples centered around node I, where a triple centered around node I is a set of two edges connected to node I. And the clustering coefficient is given by this formula 1 by n sigma. If you add all the values, that gives the clustering coefficient of the whole network. It captures the probability of the neighbors of a given node I to be linked to each other. So the whole idea is, if simplistically saying, like say three nodes, the three nodes represent me and two of my friends. The simplest idea is, if I know both of them, what is the probability that they know each other? So what is the probability that this is a complete subgraph? That is what the clustering coefficient is. Closeness centrality is basically how far away or how close are the nodes are. So uh, the farness or peripherality of a node, V is defined as the sum of the distances to all other nodes. Basically, how far are the different nodes each other, and take an inverse of that, for which you get the closeness. We standardize the closeness by dividing it by the maximum possible value, which is 1 by n minus 1. And if there is no directed path between vertex V and I, the total number of vertices used in the formula instead of the path length. And the more the central the node is, the lower is its distance to the other nodes. So intuitively thinking, it is possible because if your node is at one end of the network, the distances to the rest of the nodes are much bigger than if it isn't within the center. And it is regarded as a measure of how long it will take to spread information from that particular node to the other node. What it measures is basically the diffusibility of the network. How fast an information can diffuse through the network. That is what is measured through the this closeness centrality. Next is the betweenness centrality. It's basically an if the betweenness centrality of a node is defined as how many times it is in between the shortest path length between two other nodes. That is what it is. So it quantifies the number of times a node acts as a bridge along the shortest path between two other nodes. So you're trying to find two other nodes, you're trying to find shortest paths between them, and how many of those shortest paths pass through a particular node is the betweenness centrality of that particular node. It is a measure used for quantifying the control of a human on the communication between other humans in a social network. Yeah, this is given by, basically, this is the fraction of nodes that are fraction of paths that are passing through that particular node, and these are the total number of paths. So it's this fraction which is given by betweenness centrality. And next is random walk. So it's something that we discussed just earlier. So a random walk is known as stochastic or random process, which describes a path that consists of a succession of random steps on some mathematical space. That can be a graph, that can be a network, that can be some other things as well. And given a graph and a starting point, you select a neighbor at random, and you move to the selected neighbor, and you repeat the same process till a termination condition is verified. That you define certain termination condition. If you reach this particular node, you terminate, or if you traverse across total maybe 100 nodes, after that you terminate. Or there can be several of these termination uh, conditions. So you move randomly um, until you reach that termination condition. And the random sequence of points is selected in this way is called a random walk of the graph. And it is a method that is used to study the network vicinity of a gene protein seeds to study their functions. Just as we saw in genotype phenotype graphs as well, we use a random walk there as well. Okay, this is the concept of centrality relativity hypothesis, which states that, say, in a protein network or a gene regulatory network or a protein gene network, the nodes with higher centrality are likely to be more lethal in nature, which means if you delete that node, it is highly possible that phenotype will be lethal and the organism will not survive. uh, Something to measure this centrality lethality is by the pairwise disconnectivity index, which quantifies how crucial an individual element is for that sustaining the communication ability between connected pairs of vertices in a network that is displayed as a directed graph. Such an element might be the vertex or an edge, as well as a group of vertices or an edge or edges. The index can be viewed as a measure of topological redundancy of regulatory pathways, which disconnect different parts of the network as a measure of sensitivity of this network to the presence or absence of individual element. So here your this index is given by this formula n0 minus n of minus v by n0. This n0 is the total number of vertex pairs in the network that are connected by a path of any length in the network, and this n minus v is the number of vertex pairs that remain connected following the removal of the vertex v. So the if you remove the vertex v, how many uh, pairs of nodes remain connected? Uh, if you subtract that from your initial number of vertex pairs and normalize that by the number of vertex pairs, you get what is known as the pairwise disconnectivity index, which gives you a quantification about uh, how uh, how lethal a particular node can be. Okay. Uh, Next is diseases is network perturbations, which basically is we've gone through this before, but I'll again go through it. And cells are regulatory and signaling pathways to connect various components such as proteins, DNA, RNA, and metabolites. And this complexity allows cell to adapt to changing conditions. And the cellular networks, it's basically a network representation of biological systems, and analyzing the topology and dynamics of this network can be crucial for understanding their complex and adaptive behavior. In general, cellular networks are robust against various perturbations, but they can be extremely fragile to specific ones. A good example would be this sickle cell anemia, a single point mutation giving completely different phenotype, which is not exactly lethal, but it's close to lethal. This trade-off between robustness and fragility can lead to disease formation. Certain gene mutations or combinations can trigger cascades of failures in gene regulatory networks, which can lead to disease phenotypes. Diseases like Huntington's or sickle cell anemia are monogenic, while this monogenic meaning one gene or the the variation of one gene is responsible for the disease, but other complex diseases like cancer, diabetes, Alzheimer's, these are multigenic or polygenic. There are multiple genes involved, uh, and the various variances all add up to give you that particular phenotype. Excuse me. Okay. Okay. Next, we come to the graph topology. The first concept there is community detection. So first is what is a network community? This guy, Eric Bell, he was doing his PhD in Caltech, where uh, uh, he basically plotted his the social networks that belong to LinkedIn, Facebook, and Twitter. Uh, uh, he basically described how how communities basically arise or emerge from this particular set of set of networks, and how we can use these communities to to make specific inferences about specific things. Say, for example, uh, the, uh, he plotted his his LinkedIn, Facebook, and Twitter accounts. So you can see there is clear distinction between these colored nodes. So these is clearly distinct from the blue nodes, which is clearly distinct from the red, and it's clearly distinct from the green. But the thing is, this distinction came in naturally, and it is surprising to see that this all these red nodes are belonging to one set of his friends. These blue nodes belonging to another set of his friends, and these green nodes belonging to another set of friends. So it is this community structure is very much prevalent in across all the networks that we come across, be it biological, be it social, be it other networks, and identifying these communities can be of very good help if you're trying to say perturb or do anything with the network. Basically, you want to you know, say cure a disease, or you want to find a set of genes which are responsible for that particular disease. So there the sense of community in the network is very important. So loosely stated, this community detection is the problem of finding natural divisions of a network into groups or vertices, which are called communities or clusters, such that there are there are many edges within each group and few edges between the groups. So uh, the communities themselves should be very well connected, but one community should not be very well connected to another community. That is the whole concept. Uh, what exactly we mean by many and or few, however, is debatable, and a wide variety of different definitions have been proposed, leading to a correspondingly wide variety of different algorithms for community detection. One such algorithm is hierarchical clustering. So it requires two steps. First is the choice of the distance function. Basically, what you're trying to do is your intuition is the lower the distance between two particular nodes, the more probability is that those nodes belong to a single community. Now, how do you measure the distance between two nodes? That can be done through a number of functions. So the most simplest of them is Euclidean distance, but keep in mind this Euclidean distance is basically it's not symmetric. Basically, distance between aik to ajk, it's the same as ajk to aik in Euclidean distance, but in real life, if you're moving from one node to another, the distance from node 1 to node 2 may not be equal to the distance from node 2 to node if it is a directed network. So you have to keep that in mind. So there can be different functions. One is Euclidean. This is a cosine similarity measure. This is basically the dot product. So if you look at it, it's basically the dot product, the cos theta term, how you represent it's this. And this is the Pearson correlation coefficient, which is basically the covariances between the two elements divided by their individual sigmas or variances. So there can be various measures of these distances, but depending on mm, what kind of network you're looking at, what kind of inferences you're trying to make, you use that particular distance for your particular work. So uh, first is getting the distance function. Next is the decision of how to extend the distance function to define the distance DAB between any two clusters of nodes A and B. So not only the distances between individual nodes, you have to also calculate the distances between individual clusters as well. That's also a challenging thing to do. For that, what you do is to extend that distance function. There are several options. So first is the minimum of single linkage clustering. It's a minimum distance between the two clusters. So the minimum distance between any pairs of nodes in that cluster. Second can be maximum distances between two clusters. So you take all the nodes of two clusters and measure distances between them. The maximum distance between any two nodes of those two particular clusters is basically your complete linkage clustering or maximum distance between the two clusters. And another you can do is take the mean. So intuitively you can imagine if two clusters are represented by two circles, and the distance between the mean of one cluster to the mean of the other cluster is basically the distance between their centers. So something intuitively you can think of something like that. So hierarchical clustering depends on the distance, and also how do you calculate distance for the in in between individual nodes, and also how you calculate distance between individual clusters. These two things you need to keep in mind for hierarchical clustering. Next is the Garvan-Newman algorithm. So this if you remember, we did in quite detail. We went through a proper good example through various situations how the partition occurs. But yeah, just breeze through it. So it's an iterative algorithm, and at the beginning of each iteration, you compute the betweenness of the edges in the graph, and you remove the graph edges with the largest betweenness. If more than one edge has the largest betweenness, you remove all such competing edges at the same time. If the graph gets disconnected to two or more communities or components, we compute the total modularity score of the resulting communities. We'll come to modularity later, but yeah. So if what you do is you calculate the edge betweenness for all the edges, all the nodes in the network, and you delete that particular node which has the highest betweenness. So then what you get is two uh, if you get two individuals, uh, if you get disconnected, you will get two or more communities. If it uh, if it's get disconnected, you get two or more communities. And what you do is compute the total modularity score. And if the modularity increases compared to what it was before, then that is a valid or a good partition. If it does not, then it is not. And you repeat until there are no more edges, and the partition with the total modularity score is the optimal partition. We could stop dividing a community if the total modularity score of the undivided community is greater than the total modularity score of the divided community. Yeah. So next is the concept of modularity. And uh, the thing is, if community size is a constraint, then we are, for instance, at liberty to select the trivial divisions of the networks that pulls all the vertices in one or two groups, none in the other, which will guarantee that we will have zero intergroup edges. So your graphs will be disconnected. This division, in a sense, optimal, but it will not tell us anything of any worth. So the idea that a true community structure in the network corresponds to a statistically surprising arrangement of edges can be quantified by using a measured quantity known as modularity. The modularity is it's basically a multiplicative constant, which is the number of edges falling within groups minus the expected number of edges in an equivalent network with edges placed as random. So the idea of modularity uh, is your particular community more connected than in a random network with the same number of nodes and edges. So that's the whole idea. If it is more connected, then it is more possible that that's a community or that's a cluster, and it will have higher modularity score. And modularity measures strength of a community partition by taking into account degree distribution. Given a network with m edges, the expected number of edges between two nodes I and j, degree di and dj respectively, is given by this form, di dj by 2m. So this is your basically modularity formula. So you sum it across, and uh, you, you partition, you check the modularity score. If the modularity score after the partition is more, then it's a good partition. If it is not, then the previous partition was better. This is not a good partition. So a larger value of Q will indicate a good community structure. So yeah, now next comes the concept of network motifs. And what it is basically it's a similar concept to a community in a network. So not all subgraphs in a network will occur with equal frequency. Motifs are those subgraphs that are overrepresented compared to a randomized version of the same network. To identify motifs, you identify all subgraphs of n nodes in the network, and then you randomize this network while keeping the number of nodes, edges, and degree distribution the same. And then you identify subgraphs of all the subgraphs of n nodes in the randomized version, and the subgraphs that occur significantly more in the real network compared to the randomized one are designated as motifs. The subgraphs that are more prevalent inside your network than compared to a random network is what a network motif is. So this is one of the models to these are two models to identify network models. One is a configuration model, uh, which will generate a random graph with an arbitrary sequence of edges, and then they'll do is basically they'll cut this they'll randomly sample a pair of unconnected stubs. So basically you'll cut these edges, so what will form is these small stubs. So each edge will correspond to two stubs, and we'll randomly sample a pair of unconnected stubs and connect them, and you'll repeat till there is no stubs remaining. But what can happen is there's a possibility that a self edge or multiple edges between the same nodes or a disconnected node can also get formed. So this is the idea of getting basically a random network which you can compare with your original network to get your network motif. This is another model. Edge switching model is basically you randomly select a pair of edges, rewire them, and you keep rewiring of the net. Um, you do keep rewriting, and which is produce a randomized version of the original network. And yeah, and then you can compare uh, with your real world network and check for network motifs. So next we come to the part of systems biology. So this slide I also kept in, in the live sessions that I took then. So yeah, uh, modeling in systems biology. It's basically first you get the prior knowledge of components, interactions, pathways. Then you make the network, uh, connected network, and which things are affecting which things. And after that, keeping everything in mind, you develop basically you write ODs and you parameterize this with initial considerations and chemical equation weights and other things. And then you perform the whole simulation and you estimate the parameters, and then you try to basically check with the newer parameters if it is a better model than the other one or previous one or not. If it is a better one, then you continue. If it is not, you go back to the previous model that you had. And if the simulations do not match the experiments, then you consider this network topology, and then you change the network and do it again. So uh, once the once topology and parameters for initial conditions are set without bias, simulations should be run without altering these conditions. Parameter variation should be systematic, unbiased, and comprehensive. And topology should be altered on the basis of biological knowledge only when simulation outputs do not match the experimental observations for input-output relationships. So the network that you are forming, that should be based on only biological knowledge and literature review basically. Okay. So yeah, this is an example of how do you make these ODs. So it's for example, your X is here is a concentration of a substrate X, which can be a mRNA protein, whatever. And at time t plus delta t, uh, you can expand this into a Taylor series. So X of t plus delta t is, if you expand in Taylor series, it looks something like this. And if you take the limit of delta t tends to 0, it is what gives you the differential equation form. So delta t tends to 0, x of t plus delta t minus xt by delta t, which is basically dx by dt. So this dx by dt is, this k here is the um, chemical reaction, the reaction constant or the reaction rate. So dx dt is equal to kx. So this is a first order linear autonomous ordinary differential equation. It involves two quantities. One is basically your x, which is your state variable, and your k is the parameter. It describes the reaction rate. That is how the concentration of X changes with time. So this is how you write ODEs using biological information. So after you construct the ODEs, you need to estimate the parameters of the model. So uh, to ensure the accuracy of the mathematical model, model parameters must be estimated using experimental data, a process called regression. Also, pre-regression and post-regression diagnosis must be employed to evaluate the model goodness of fit and reliability of the estimated parameter values. One such technique to do parameter estimation is MLE, which is maximum likelihood estimation. And another one is least square fitting. So these yield parameter values and the variance covariance matrix. Uh, they work on the assumption that the estimated parameters have a normal distribution. When this assumption is not valid, Bayesian inference can be used, yielding the full parameter distribution. Uh, Parameters in probabilistic models must be inferred by either indirect inference or Bayesian methods. In indirect inference, model parameters are estimated by minimizing the differences between intermediate statistics and characterize simulated and experimental data. So we have talked a lot about parameter estimation and how these processes work. I'm not going into detail, but this is a boring thing to do, but this is one of the most important things for the model. But yeah, okay. Next we come for evolutionary algorithms. Yeah. Uh, so yeah. So first is the concept of evolutionary computation, which basically integrates the concepts of biological evolution into algorithms for solving complex optimization and design problems. Examples include sequence alignment, phylogenetic inference, drug design, etc. So the questions that evolutionary algorithms can answer is how quickly do well-adapted individuals emerge, or what genomic organization strategies influence adaptation, which characteristics of evolved objects are outcomes of the evolution process, or how do population structures and selection mechanisms impact diversity. So it's basically a computational equivalent of Darwin's evolution theory. And yeah, okay, I think, okay, now this is a population. I have not uh, kept the other slides. So uh, there are two components for creating a genetic algorithm. One is a population and another is a fitness function. So they these algorithms use components of fitness, inheritance, natural variation through recombination and mutation to model how a genetic lineage will adapt over time. Therefore, these components must also be incorporated into the genetic algorithm in order to more accurately mimic natural selection. So, the first is the population. The population is defined using the first generation of F1. This can be a set of genes or other units of interest. This generation can be represented in different ways, but the most common technique uh, is to have a bit array with different permutations and zeros and ones. What this sorry, what this actually means is, so this is a 2D bit array. Each of the rows will correspond to an individual chromosome, and each of the individual chromosome, what it in the modeling sense correspond to is a set of parameter values or a set of your solutions. And each of these solution will be a particular gene. It can be either 0 or 1, or in this case, it can be either 0 or 1 because this is a bit array. So, so this is your structure. So this whole thing is your population. Individual rows are your individual chromosomes or a set of values, and each value is the gene. So this is how the population structure looks like. And how does the genetic algorithm work? So you have some starting point where you generate a random population of n chromosomes, and then you fit evaluate the fitness of each chromosome X in that position. So how do you calculate that fitness? You define a fitness function, or basically an optimization function, which you try to maximize, and you calculate the fitness of each of the components, each of the chromosome components, and uh, uh, mm, after that you do what you do is evolutionary concepts like crossing over, selection, mutation, survival selection, and then you iterate over this. So first you take a population, you calculate its fitness function. Then what you do is crossover. You just randomly swap out. If you remember, I don't know if you remember or not, I showed you some slides. So there can be different kind of mutations. So uh, you can directly swap out. You can I, I don't think I have it open here, but yeah, you can different have different kind of crossovers and mutations. So between any two chromosomes inside the population, and you check if it is if it is good enough for selection, and then how do you do it? You do fitness calculation. If it is good enough, uh, uh, if it has a good enough fitness score, then it is a good enough uh, result. If it does not have, then you need to improve the fitness score again, and maybe you have to try another set of crossovers or mutations. So what does uh, hmm, selection mean here? So select two parents of the chromosomes from a population according to their fitness. The better the fitness, the bigger is the chance to getting selected. Any two set of chromosomes. Now you cross over those. So what is this crossover? We there will be some probability associated with it. So uh, uh, if basically you consider these two individual chromosomes as parents, and you cross over individual values to them, and then you check for mutations. So You can do only crossover, or you can do crossover and mutation. So this range of your parameters will basically consist of an individual chromosome. So your individual chromosome is considered a set of parameters, and another chromosome will also have a set of parameters. So when you crossover between them, then you mutate or you do not, depending on your algorithm, and then you check for fitness scores uh, according to your optimization equation or problem. And then if it passes a certain threshold that it is a good enough solution for the problem, if it does not, then you need to do this iterate again and again. So, and there should be a termination condition. So, if it reaches a certain point or crosses a certain threshold, it should be terminated. Next, we come to the concept of metabolic networks. So, these are comprised of chemical reactions which involve metabolites, catalytic proteins, and metabolic pathways, etc., etc. So, the two types of metabolism: anabolism and catabolism. Anabolism is for synthesis; catabolism is for degradation or breaking down. And this is how a metabolic network looks like. And for the simulation of these networks, the most important thing is the knowledge of the reaction kinetics. These kinetics entail concentration gradients of molecular partners relative to initial concentrations. And yeah, this is how basically your metabolic networks look like. And this is how you reconstruct you can construct metabolic networks using different databases, different literatures, and everything. And the components of reconstruction include, uh, for simple graph analysis, these nodes which are metabolites and these interactions which are basically depicting reactions, these are needed. If you do want to do further flux balance analysis, the stoichiometry of the reactions and flux information, which include maximum upgrade rates, etc., etc. These are growth rate, etc. Those are required, and you can have the whole metabolic network. So next comes the concept of stoichiometric matrix. So the stoichiometric coefficients denote the proportion of the substrate and product molecules involved in a reaction. For example, for a reaction, if you consider this reaction, A plus B equal to 2C, uh, the stoichiometry for A is 1, A is minus 1, B is minus 1, and C is plus 2. Now, let's assume that our metabolic network has, say, for example, R reactions, R1 to R. And if you consider this reaction Ri with operating with uh, rate Vi, then your rate vector will look your rate vector, which is given by this V, will consist of all the dates of those particular reactions. So each row will correspond to a particular reaction, and in your column will have all the dates as a vector. Now, similarly, you can do for the concentration vector, you can have a list, basically a vector of all the concentrations of X corresponding to the particular dates. And what you do after that is basically take it to this form. So dxi by dt should be STV. So this ST is basically a transpose of your stoichiometric matrix, and uh, this is the form that they are expressed in. So dxdt equal to S into V. So V is the vector of your rates, reaction rates. S is your stoichiometric matrix, and X is basically your gene or metabolite of interest. So, for most applications of stoichiometric matrix, assume that the system is in so-called steady state. So, what is steady state? It is that state in which the concentrations of metabolites will remain constant over time. That's the derivative of this concentration is zero. So basically, in steady state, your x will not change with t, so your dxdt will be zero. So which implies if you sum the product of your stoichiometric matrix and your rate vector across all the reactions, that should be equal to zero. So this is a very important equation that we have used and we use all the time. So we assume steady state and we show that it's equal to zero, and we like try to find rates of different reaction or maybe stoichiometry of a particular compound, whatever you're trying to find, whatever is missing, you can find it using steady state approximation. Okay. Next comes the concept of constraint-based modeling. Uh, it is based on the assumption that organisms exist in particular environments that typically have scarce resources, and over time, the probability that the fit will survive is higher compared to the probability that the survival of the lesser fit. To be fit for survival, a myriad of constraints must be satisfied, which limits the range of available phenotypes. So these constraints can be divided into certain categories. So for first type of constraints is physiochemical constraints, which are mass balance, energy balance, momentum balance, etc., etc., etc. Next comes your topobiological constraints. So for example, steady rains inside the cell, or any kind of three-dimensional constraints inside the cell that does not allow a particular reaction, that does not allow a particular interaction, or it may specifically allow for that particular interaction depending on your network and model. Um, the other type of constraints are environmental constraints, which are basically nutrient availability, pH, temperature, etc., etc., and your regulatory constraints. So these are these are these are not technically hard constraints, but they are implemented in the cell in various ways, including the amount of gene products made, which can be done through transcriptional or translational regulation, or even post-transcriptional regulation, and enzyme regulation. So there can be several regulatory constraints. So in the first few modeling lectures, if you remember correctly, we did not account for these regulatory constraints. In the later stages, when we did RFPA, we accounted for this regulatory constraints. So these are the different constraint based different constraints that we can use for this constraint based model. Okay, the first is this flux balance analysis for constraint modeling. So this is the whole idea. So first, if you rate a set of metabolic reactions, formulate the stoichiometric matrix, then you apply mass balance constraints, basically your SV equal to 0, a steady state approximation that we define. And then we define this objective function, uh, which will calculate uh, a certain score for for that particular um, solution, and you need to optimize this set. So you need to either maximize or minimize this function depending on your function. So what you'll get is a set of, so your solutions will look like this. You'll not have a single solution. What you'll have is a solution space, which will be defined by these constraints or these boundaries. And your optimal point should be the point that will maximize your objective function. So in this case, this is this point. So first you build the metabolic network, and then you make the stoichiometric. Basically, you convert this reconstruction into a mathematical model by forming the stoichiometric matrix, where each row will represent a metabolite and each column represents a reaction. Then you do steady state approximation. At steady state, it will be the flux through each reaction is given by SV equal to 0. And this is the thing, that since there are more reactions than metabolites in large models, there is more than one possible solution to this equation. So number of reactions will be much more. Basically, the number of uh, columns will be much more than the number of rows. So it will be kind of a fat matrix. So what we'll have is a set of solutions, not a single solution. So there comes the involvability or function of this objective function, which is given by z equal to CTV in this case, where C is a vector of weights indicating how much each reaction contributes to the objective function. And in each in practice, when only one reaction is desired for maximization or minimization, C is a vector of zeros with a 1 at the position of the reaction of interest. While simulating growth, the objective function will have a 1 at the position of the biomass reaction. And then you can check for, you have the solution space something like this. These are your constraints, bound by your constraints. So the point at which the objective function is maximized, that is your optimal solution. Uh, yeah, finally, LPP can be used to identify a particular flux distribution that maximizes or minimizes this objective function while observing the constants imposed by the mass balance equations and the reaction terms. Simple enough. Flux balance analysis. So this is basically, this slide is showing the importance of uh, objective functions. So the simulation results indicate that the choice of the objective function will significantly impact replicative lifespan and generation times. So this is an experiment that was this is an FPA model that was simulated on basically E. coli, and they basically the objective functions were different in different cases. So one was maximal growth, another was minimal glucose uptake, different types of objective functions they were testing. And after the simulation results, they found that this choice of objective function is significantly impacting the replicative lifespan and generation times. Cells without maximal growth optimization showed limited growth, while some failing to divide or doing so infrequently with longer generation times, emphasizing the importance of maximal growth in replicative aging. So the importance of identifying and using the correct optimization function is very important, because your whole model will depend on it. So you need to understand the biological system, and you need to uh, provide that objective function in a much more soft, like, what to say, much more educated manner, much more like you should know, okay, like at which point of time the system should maximize for ADPH, or at which point of time the system should maximize or minimize ATP production. So those kind of domain knowledges are very important to construct that particular objective function and simulate model. So this is a variation of flux balance analysis, which is called MoMA, minimization of metabolic adjustment. Uh, it's an algorithm for predicting metabolic flux distributions after a gene knockout. So, uh, although the assumption of optimality for a wild type organism may be justifiable, the same argument may not be valid for genetically engineered knockouts that were not exposed to long-term evolutionary pressures. So, uh, thinking of the objective function, it is not more and not anymore sufficient just to maximize your objective function, because in a gene knockout scenario, it'll be the scenario may be different. It may be lethal. It may not be valid for genetically engineered knockouts as well. So depends. In the basis of this method, lays the assumption that after a knockout, metabolic fluxes undergo minimal changes. So here comes the concept of robustness as well. So metabolic fluxes undergo minimal changes with respect to the flux configuration of the wild type, and MoMA employs quadratic programming to identify a point in flux space which is closest to the wild type point, based on the conjecture that the mutant remains initially as close as possible to the wild type optimum in terms of flux values. So uh, it calculates this is basically the Euclidean distance between the flux vector v and a given vector w. So basically, what it is trying to do is, this is your feasible space of wild type, which is given by this. So uh, given by this green part, and this is the feasible space for the knockout, which is given by this yellow part. So what MoMA will do is, it will try to minimize the distance between this wild type and so this knockout and the wild type. So it will calculate pairwise distance values from this knockout boundary to the wild type boundary, and whichever point is the closest, the least distance, that is the optimal point according to MoMA. Okay. Next is a similar concept of sorry, of room, which is basically regulatory on of minimization. Uh, the key difference between MoMA and room is basically incorporating regulatory dynamics, incorporating regulatory constants. These are the major differences between MoMA and room, that it incorporates these regulatory uh, mechanisms inside the model. So it acknowledges that genetic regulatory changes post gene knockouts are minimized by the cell to reduce adaptation costs. It operates under the assumption that the regulatory changes can be parsimoniously described by Boolean on of dynamics, assigning a fixed cost to the each of the regulatory change, irrespective of the magnitude. This is some assumption that they make, which is actually not very true, but in this case, it works out well. So, uh, and also, it also incorporates implicit regulatory concepts. Unlike MoMA, which does not explicitly incorporate regulatory constraints, Room implicitly accounts for regulatory changes by identifying significant flux changes in metabolic reactions. This is essential as efficient gene expression, as efficient expression likely requires adjustments in flux implicating changes in gene expression rates. So the major difference between MoMA and Room is this incorporation of this regulatory mechanisms. So yeah, these are different predictions. So this is given by this is a flux distribution. This B is MoMA's prediction, and C is Room's prediction. So in all the yeah, in Room prediction, the linearity of the flow is preserved in branch point B. So this point it's preserved as opposed to MoMA's prediction. So this is Room's prediction. So this is preserved as opposed to this, because like this is still v2 equal to 5. This was still, and now it is still 5. But this is changed here in case of MoMA's prediction. Uh, the MoMA solution has a Euclidean distance from 62.8 from Room solution and has three altered fluxes with respect to this original model or original network. On the other hand, MoMA solution has 9 altered fluxes with respect to A, and Room solution has a Euclidean distance of 75 from A. So, see, the MoMA was trying to minimize the Euclidean distance from its wild type solution space to its knockout solution space. So, the, the Euclidean distance is lower here, while Room did not try that, so it is much higher here. So, yeah, this is an example difference of how both of them work. Okay, so next comes the concepts of gene protein reactions. So, uh, gene protein associations, you say. Uh, this is a simple association where a single gene is influencing a protein which is influencing a reaction, or it can be of this form as well. So, these are what are known as isozymes. So these are enzymes that catalyze the same reaction through different proteins. So these are different enzymes, these are different proteins, but they're catalyzing the same reaction. So that it is what is known as isozymes. So enzymes that are similar in function. They catabolize, they metabolize the same reaction. So, enzymatically metabolize the same reaction. Yeah. So yeah, this third panel is showing multimeric protein complex. So basically, two genes are contributing to a protein. This may be a dimer, this may be a tetramer, whatever be it. But the components of this protein are coming from two genes. So these are known as multimeric protein complexes, and this protein goes and affects the reaction. Or it can be of this form as well, multifunctional. So one gene giving one protein. Now this protein is involved in two reactions. So this is where redundancies come from, right? So this is where redundancies coming from. So isozymes giving one reaction. This is one protein giving influencing two reactions. Okay. So this is another concept in basically uh, when you're trying to metabolic models only. A dead end metabolite, it is defined as a metabolite that is produced by a known metabolic reactions of an organism, but it has no reaction consuming it inside the system, or that uh, basically it is static within the system and it's not being able to move outside, or it is not getting consumed inside. So these dendritic metabolites are isolated compounds within a metabolic network. In some cases, this dendritic metabolite reflect a deficit or an error in how metabolic database represents knowledge from scientific literature. In other cases, the systems level analysis alerts us to areas where more experimental research is required because you do not know what is happening. So in this case, uh, the compounds A and C here, A and C, are neither getting produced nor getting transported from the system. So these are examples of dead-end metabolites here. So next is the blocked reactions. So initial metabolic network reconstructions often contain inconsistencies due to annotation errors or unknown enzyme functions. So they can lead to missing reactions in a draft model, creating gaps in metabolic pathways. So these are dead-end metabolites, and these are classified into two groups: this root non-produced and this root non-consumed. So this is non-produced. Basically, the system is not producing that, but it is still involved in the system. And another is root non-consumed, is your system is not consuming that particular element or the particular compound or the gene, but it is producing it. So they have divided into RNC and RNP. So this RNP RNC block reactions cause further gaps downstream or upstream. So which are downstream is known as DNP and upstream UNC. And identifying these consistencies, which are these dead end metabolites, is known as the gap finding problem. So so this we discussed in detail, great detail, but yeah, I'll go through it again. Flux variability analysis. So how is it different from FPA? It basically what happens is FPA predicts steady state fluxes of reaction in metabolic network based on biological imperatives. Now FPA doesn't always yield a unique solution, which is why this flux variability analysis is required, because it quantifies the feasible ranges of reaction fluxes at optimal or suboptimal production rates, production states determined by FP. Uh, Flux variability analysis is a generalization of FPA, and it aids in understanding metabolic network flexibility. Applications of these include medicine, health, etc., etc. Uh, the idea is to firstly identify the maximum value of the objective function and constrain objective function to this value. Then what you do is minimize and maximize each flux independently to identify flexibility in the fluxes across the alternate optima. So this is your variability, this is your alternate optima, and what you try to finding is this variability of the fluxes in an alternate optima. So you can define a range of values. Uh, so it's basically it's minimizing and maximizing each flux independently to identify the flexibility in the flux. So you want to define a range of those flux values where it can still be giving the optimal objective function value. So if you have n fluxes in general, you have around 2n FPA probably. Okay. Yeah. So uh, this is basically FVA in a nutshell. So what you have is this feasibility region, which is the maximum and the minimum. Uh, your objective is to your I is the metabolites and J is the reactions, and what your objective is to maximize and minimize VJ for all IJ. So you want to find the minimum and the maximum value. So yeah, you need to solve this optimization problem, and what will give you it is a range of in which those fluxes can reside, which can still give you the optimal solutions. So we looked at knockouts. Uh, Now we'll look at overexpression and synthetic lethals. So overexpression algorithms, it's a simple framework, FSCOF. It searches for the candidate fluxes to be amplified, scanning through for those fluxes that increase with enforced objective flux. So uh, the basically FS objective flux under the objective function for maximizing biomass production flux. Uh, During FSCOF implementation, three types of intracellular flux profiles are typically identified: increased, decreased, and unchanged. Oscillating flux profiles are found to be rare in case. So what is happening is, this is your initial product. This you're trying to enforce the objective flux of the product formation and trying to increase it to the Vmax, and Yeah, what you're trying to check is basically how your biomass formation is increasing or decreasing. So this is how overexpression algorithms work. So it'll you have a certain initial product and an initial expression, and then you enforce the objective flux of the product formation. So this is your constant. So you're increasing your uh, objective flux for the product formation and check for basically decreased, increased, or unchanged flux. And uh, basically, uh, u- using the biomass, you're trying to maximize some objective function. Okay. Now, during FSCF implementation, three types of intracellular flux are typically identified: increased, decreased, and unchanged. Oscillating flux profiles are rare in nature. Among them, FSCOF identifies the fluxes showing the increased profile as the primary amplification target. So what you're trying to do is you're trying to increase or overexpress a particular product. So you're checking to the objective function. Uh, if you increase your objective function, is your flux increasing or is your flux decreasing or is it unchanged? If it is increasing, then that overexpression is working. Then you can uh, like you can have a good result basically. Okay, so yeah, the next is the concept of synthetic lethals. So synthetic here means it's the combination of entities to form something new. So classical synthetic lethal occurs when inhibition of two genes is lethal, while inhibition of either gene is not. So you have two genes. If you inhibit both of them together, then maybe the cell dies. But if you inhibit either of them, the cell is fine. It can be harnessed to treat diseases like cancer by selectively identifying inactive genes and targeting their synthetic lethal partners. In accordance to this concept, synthetic lethals can be applied to genes and small molecules used to elucidate the mode of action of drugs. Uh, the principle of synthetic lethal in cancer is basically loss or inhibition of either of the protein products of gene A or B. So this is gene A or B, gene A and gene B. So either of the protein products, if it is lost or inhibited, uh, uh, basically, if you do either of the inhibition, it's to heal. However, the sorry, I think some network issue. I'm still audible, right?
Yeah, you're audible.
Okay, I think there's some network issue. Okay, yeah, yeah. So now, pharmacological intervention by the partner gene product will result in synthetic lethal interaction in tumor cells with a loss of function mutation in the tumor suppressor gene. So what will happen is basically you have identified one gene. Uh, you've identified basically a set of genes which if you try to inhibit or suppress them, maybe the cell does not grow or the cell dies. But in this case, what they are doing here is basically you have this gene A and gene B. It is giving uh, in case of synthetic lethality in a single gene. There's a mutation which is basically converting it. It is basically converting it into a tumor cell. Now Now see, this gene B is already mutated in your tumor cell, but your gene A is not. But these two together are synthetic lethals. Now, if you want to kill the cell, your gene B is already mutated. You just need to suppress gene A. If you suppress gene A, then he'll be lethal because both the functions are gone. So that's the idea that they use for cancer uh, drug discovery as well. So this concept of synthetic lethals is you have a set of genes together that are together, but if you suppress all of them together, the cell dies. But in this case, this set of genes are they individually are contributing to the cancer. There's maybe one gene left. There's one gene which is still not mutated, but the other gene is mutated, which is contributing to cancer. Now, if you want to kill the cancer cell, you need to inhibit both these genes together. Now, the second gene is already inhibited because or mutated, whatever it is not performing the same function. Now, you just need to intervene and inhibit this gene A, then the synthetic lethal phenotype is getting formed. So, yeah. Next is the concept of RFPA. It's just the same as FBA, just include the regulatory component here. So this there are transcription factors and genes and proteins involved, which basically regulate these metabolic networks. So RFPA predicts regulatory and metabolic steady states for short time intervals. It computes regulatory states consistent with the previous metabolic steady state, and it utilizes FPA to find flux distributions consistent with current regulatory states. And it offers a glimpse into cellular behavior, for example, E. coli growth, but there are some limitations. So, pro, applying sorry, I think this is wrong. Yeah, applying RFPF in the study of E. coli, uh, there was a study where they analyzed the effects of metabolic and regulatory constraints on metabolic behavior. While metabolic constraints were found to determine the flux activity state of a majority of the genes, the role of transcription regulation was also found to play a role in determining the state of over 13 to 20% of the genes. So not only one layer of regulation, there can be multiple of layers of this regulation, which is very harder to model in case of FPA and its extending its extension of RFPA. So this is how integrated metabolic and regulatory networks will look like. So you have set of transcription factors which will be controlling genes, which will be controlling enzymes, which in turn will control this metabolic network or metabolic reactions. Yeah, so this is your whole concept. So you have a start, initial conditions, you have system states, and what you do is you basically you move from here to the, the regulation is given by boolean uh, boolean values. It's either on, off, or whatever it is, and then you put constraints on that, and you do FPA optimization. Then it's the same iterative process. You move on and on until you reach a ending criteria or you find the optimal number of solutions. But the major difference here is this incorporation of this Boolean expressions to simulate this ODE model, which are the basically regulatory constraints. Uh, the concept is generalized to integrate FPA, also incorporating ordinary differential equations to simulate regulation. The algorithm is an iterative procedure generating time series output at each iteration. Okay, so next we come to the last part of the modeling. So we have logical models and Boolean models. These are the last things we're going to talk about. So uh, this logical models are much more simplistic than OD and OD models. So this is the most basic and simplest modeling methodology. They represent these logical models represents the local state of each entity in the system, which includes genes, proteins, and small molecules at any time as a discrete level, and the temporal development of the system is often assumed to occur in synchronous, discrete, and time steps. Entity levels are updated at each step according to the regulatory functions. So here, these are your regulatory functions. You are defining certain relationship between these genes A, B, and C. So if, say for example, this is a no, the second one is an or function. So either of A or C will give a true B. So these kind of regulatory functions using logical models you can do. So and there are two types of these logical models. One is the one that I just talked about is the Boolean networks, and the other is the Petri net. So in Boolean networks, it'll be either 0 or 1, transitioning accordingly. And in PetriNet, uh, this, this consists of entities and transitions connected by these arcs. You see, these are transitions T1, T2. These are entities, and these are connected by arcs. And basically, transitions consume tokens from input places and produce tokens in output places while firing. So it's a similar concept, but it's slightly different because we use in Boolean network we use Boolean variables, and here we use a different kind of different kind of entity called tokens. I'll not go into the detail, but yeah, this is what logical models are about. So yeah, one type of it is Boolean models. So Boolean networks are common qualitative modeling framework for gene regulatory networks. These are capable of describing various biological phenomena which include oscillating, multi-stationarity, switch-like behavior, etc., etc. So this is if you look through literature of systems biology, you find a lot of work that focuses on transitions or multi-stability or bistability, this kind of stuff. They always use Boolean models because these are ideal to predict those things. So in a Boolean network, uh, your Boolean variables will represent gene activation states, which is one for active, zero for inactive. This your Boolean functions, which is the basically the relationship between the two the three elements here. These are the Boolean functions. These Boolean functions determine the state of each variable based on the states of other variables, and uh, the, this can be shown as directed graphs where nodes represent Boolean variables and edges will denote the influence of one variable on another, and promotion and inhibition also information also you can add. So yeah, so ultimately we come to robustness in biological systems. So we have talked a lot about robustness today anyway in the beginning, but yeah, just to give an overview, uh, it is considered to be a fundamental feature of a complex evolvable system. It is attained by several underlying principles that are universal to both biological organisms and sophisticated engineering systems. Robustness facilitates evolvability, and robust traits are often selected by evolution. Such a mutually beneficial process is made possible by specific architectural features observed in robust systems. So, what it is meaning is if you pull a particular uh, system out of its steady state and into some other state. Say, for example, if this is your steady state, you move the system here, and if it is going back to the original steady state, it is robust in nature. It is what is known as a robust adaptation. So now this can also happen if you have moved the state so much out of its initial attractor state, it is getting attracted to a different state. So you can transition to a new attractor as well. So if it is coming back to its old state, that's a robust adaptation. If it is not, if it's moving to another state, it is not. So uh, certainly there are trade-offs between robustness, fragility, performance, and resource demands. But insights into these inherent properties of robust systems provide us with a better understanding of complex diseases and a guiding principle for therapy design. Yeah. And how does this robustness work? So there are three different conditions that I've discussed. I discussed this in the last class as well. So first is the concept of redundancy and diversity, which is basically a fail-safe option. If one gene is knocked out, there's another gene which can take up its action. Um, the concept of alternative or fail-safe mechanisms enhances robustness by providing multiple means to achieve a specific function, meaning same genes or same proteins doing the same function. An example would be isozymes. This mechanism encompasses redundancy, diversity, and overlapping functions, offering flexibility and adaptability. Gene duplication followed by specialization and extensive gene loss serves as a crucial mechanism for evolutionary innovation. And at network level, examples like glycolysis, oxidative phosphorylation demonstrate alternative pathways adapting to environmental changes, showing phenotypic plasticity as part of robustness. So if you look at these metabolic pathways, glycolysis, oxidative phosphorylation, these have many intermediate, also other pathways like gluconeogenesis, fatty acid metabolism, amino acid biosynthesis. These several of these pathways, these metabolic pathways, be it anabolism or catabolic pathways, whatever it is, they have a lot of common intermediates which they use in the particular cycle or in the metabolic reaction. So a same compound or same entity is getting used in different pathways, different metabolism. So this is what brings about the redundancy or the fail-safe option. Basically, if you can have a single gene which is involved in multiple things, if you knock that gene out, it is highly possible that uh, the system will not be robust. So there comes the concept of decoupling and modularity. So the concept of decoupling in biological systems serves to isolate these low-level variations from high-level functionalities, thereby providing robustness against perturbations. And examples such as molecular chaperones like HSP90, which not only correct misfolded proteins but also buffer genetic variations, allowing for adaptation while maintaining genetic diversity. Feedback controls, as seen in bacterial chemotaxis and Drosophila, compensate for changes in network interactions on initial states, mitigating the impact of mutations. And the last concept here is modularity. It's basically we just discussed modularity a little while back, but in this case, what it does is basically it serves as an effective mechanism for containing perturbations and minimizing their effects on the whole system. It encompasses physical, functional, spatial, and temporal modules, each contributing to the system's robustness and versatility. Examples include cells, which form basic units of the multicellular organisms and interact with their environment and other cells, as well as hierarchically organized structures such as tissues and organs. So there should be a clear division. There should be clear modularity. So each of those modules can contribute to the robustness. Logical modules, on the other hand, are less overt and are equally significant, as demonstrated by systems such as segmental polarity networks. The bacterial flagellum and its control module exemplify both physical and logical modularity, highlighting the adaptability and resilience afforded by modular organization in biological systems. Yeah, I think that's it for the course. Yeah, I think this little recap of the whole course helped. I purposefully did not take that extra class because like only two or three of you attend, so there's no point in taking another whole class. So that's why I thought, okay, I'll cover uh, the major topics that I covered through these 12 weeks in one go, so that you guys can have a brush up of the older concepts. But even after this, if you have any problem, do not hesitate to write to me in my mail. I'll just give the mail ID once again because this is the last time I'll take this class. So yeah, if you have any problem, do write a mail to me. And
Actually, this mail ID, and then I sort of went to the IAC website and then took your mail ID, but I couldn't send you a mail. Like, I mean, uh, there's some error popping up. Uh,
okay, I have another mail ID.
yeah.
So I'll give you my personal mail ID as well. So I have two mail IDs of ISC. So these are the
Ah,
two,
okay. Yeah.
and I have
Okay.
another personal mail ID if you want. I'll give that as well. Yeah.
Yeah. Thank you. Because I use that, and then so
But
Google showed this big default deliver incomplete mail. So
oh, okay.
there's temporary problem, and then it will retry in 47 hours, and then basically it sort of failed to deliver the email. So
Oh,
I
no
didn't.
problem. You can use any of
Yeah,
the other mail IDs as well.
yeah,
Just
yeah.
write a
Thank
mail.
you.
Okay. Okay.
Anyway, yeah, thank you for covering the whole like brushing everything up. It might have been a
Yeah.
bit of a task for you as well.
Yeah. You guys are all appearing for the exam, right? Both of you.
Yeah, yeah.
So when is the exam?
27th.
27th of April.
Yeah, yeah.
Okay. Okay. Cool. So you around. What 2 weeks, around 10, 12 days you have left, right?
Yeah, yeah.
Okay. Best of luck for your exams. And if you have any
Thank you.
problem, any issues, just write it to me. I'll do my best
Yeah,
to help you out. Okay.
yeah, yeah. Thank you.
And thank you for joining the classes. And uh, thank you
Thank you
for
for
being in the class. Yeah.
So thank you
teaching
so
us
much,
to,
sir,
yeah.
for conducting this 12 weeks great session.
Sorry, can't hear you.
Actually, I can't able to follow up much more because as I am BTech student, because I will come by only like 6:30 to my home. But I am, but I have tried my best to follow up. Sir. But
Yeah, yeah, sure, sure, sure.
yet, yet there are many things to be covered in the courses. Like I will get many doubts because I can't follow up the course exactly.
Yeah,
But I will try.
it's I can understand for a first time someone who is taking this course, or first time someone who is reading about all this, it's a little bit difficult. It's understandable.
Yes, sir.
But yeah, if you have any doubts, do write me a mail. I'll be happy to help.
Yes, yes, sure, sure. Thank you so much.
Thank you.
Thank you,
Thank you.
guys. Thank you for joining the classes, and best of luck for your exams and in your future in
Sure.
general. Thank you.
Thank you so
Thank
much.
you.
Thank you.